So while we're getting all set up here, this symposium came together in the most interesting way. I had no idea 5 weeks ago what this was going to be about, and I was contacting the most random people. A man who, put out a paper on evolutionary genetics of the ADSL gene between Neanderthals and modern humans. Thank you. And and then right at the end, in the last 5, 6 weeks, we've had three big papers come out. And so it was just,
Mhm.
I was able to loop you all in. Dr. Manoj is one of those who has a really interesting and exciting hypothesis. And so we are excited to hear what you have to say. Are you ready to share your screen?
Yes. So my work basically defines the role of complement in induction of disease pathology of ADSL deficiency disorder. Recently, I came in this field. You see this recent publication, and very recently we generated some preliminary data, and that suggests complement is playing important role in this disease. So this slide shows information about the complement activation pathway. Complement activation occur mainly through three pathways, which is called the classical, lectin, and alternative pathway. So classical pathway of complement activation occur when IgG antibodies or IgM antibodies to certain antigen make the immune complexes. And these immune complexes, when recognized by their receptors on variety of immunological or neurological cells, they causes the complement activation. Similarly, the lectin pathway happens because of the recognition of lectin or phycolin to their receptor, mainly the Manoj receptor, like my name, Manoj receptor, and that causes the complement activation. And the third pathway is the alternative pathway of complement activation. This is interesting. This pathway activation occur spontaneously. This basically what happened here when certain injury or the cell death happen that causes the hydroxylation of the complement, and that creates the molecule, is the enzyme called C3 convertase and C5 convertases. So downstream of these convertases is the production of, causes the downstream cleavage of complement 3 or complement 5, causes the production of C3A and C5A. So C5A is the terminal complement activation product, which are known for developing neurodegeneration in certain neurological diseases like Alzheimer, Parkinson, uh, Huntington, and many other diseases. Do you guys see my slide properly, right? Okay, perfect. So this slide shows information about the complement 5A. Complement 5A is the protein fragment of 11 kilodalton, which which exhibit their pro-inflammatory factor function only when it bind with their C5AR1 receptor. So there are two receptors for this molecule, C5AR1 and C5AR2. C5AR2 ligation is known for not causing inflammation, but C5AR1 is. So pretty much my talk will be focused on C5AR1. So C5AR1 is the member of large family of G protein coupled receptor, which is found to expressed on variety of immunological and neurological cells. So in immune cells like macrophage, monocyte, dendritic cell, neutrophil, they all have C5AR1. In brain cells, microglia, neuron, oligodendrocyte, and astrocytes are known for having its expression. And in this slide, I do not need to tell anything, you guys, because you all are expert in this area, but just for the fresher. So ADSLD is basically happened because of the deficiency of ADSL lyase enzyme. The major function of this enzyme is to catalyze SAICAR to AICAR and SAMP to AMP, which is important for purine metabolism. However, when a ADSL defect and the resulting deficiency of ADSL happen, this function is not working, means SAICAR is not able to convert into the AICAR, or SAMP is not converted to AMP, and then these molecules stay inside these cells. And because of this deficiency, they convert into their dephosphorylated form, which is called the SAICAR and S-ADO. And it has been known that this ratio is important because it correlates with the disease severity. So basically, if SAICAR is higher than S-ADO, it has been seen in more severe cases of ADSLD disorder. So I come up with this hypothesis, and this hypothesis is that because of the ADSL defect and resulting ADSL enzyme deficiency, the proper degradation of SAICAR is not happening, and it is converting into the dephosphorylated form. And this is toxic for these cells. So it causes the cell or tissue injury. And this injury takes the third pathway of the complement activation, which is called the alternative pathway, where it causes the spontaneous hydrolysis of complement 3 and making two enzyme, C3 convertase and C5 convertase. And that causes the downstream cleavage of C3 and C5 to produce C5a, terminal component. So this is the hypothesis. And now here you see when SAICAR is in normal conditions, SAICAR should convert into the AICAR. But because of this deficiency, this thing is not working properly. And AICAR is known for, AICAR actually act as an anti-inflammatory component. So AICAR is known for inhibiting the complement factor B, and that actually involved in the alternative complement pathway. So this actually proved this hypothesis even one step more clear, why that would be reason that complement could play important role here. To prove this hypothesis, so we were not in a stage to generate data. And after this paper came and I got the call from Nicole, and I also got call from the Emma and Priya. So I was thinking, all tools are ready in my laboratory. Let us see something we can do. So what we did, we were generating the microglial cells from the iPSC. We were generating the dopaminergic neurons from the iPSC. So we had the controlled human iPSC. And we generated microglial cells, and we confirmed these are microglial cells. So microglial cells, as you know, are having the positivity of the CD11B and TMEM119, which is more specific marker for the microglial cells. So the microglial cell that we generated from the iPSC of normal human is actually microglial cells because they are showing their marker. Similarly, we use the neuron developed from the iPSC, and we tested them for positivity of the TOJ1 and MAC2, and we have seen, as compared to isotype, you don't see anything, which is a good thing because that is showing our neurons that we generated is actually the neuron. So the purpose was generating these iPSC-derived microglial cells and neuron was to perform co-culture experiment. So we first developed the cell model of this disease. I come up with the idea because what is happening in this disease, ADSL deficiency. So how we can mimic ADSL loss in our cell system. So I come up with idea to use the monoclonal antibodies to ADSL, which is available. So we treated these cells both in the presence and absence of ADSL antibodies for 12 hours. And the first thing we did, we just take images of this cell culture. So you see on your left, this is the control cell where you see these neuronal cells are giving you the proper structure of the neuron. So you can see the dendrite, right? You see the bigger size. And interestingly, in the disease cells, they lose their structure significantly. It looks like they are dying. So then we wanted to know whether complement is playing some role here. So what we did, we used these cells and measure C5 and C5a in both the control cells and the disease cells. So what you see here, as compared to control, disease cells are showing significant increased expression of C5 and C5a. So as I said, C5a is playing a destructive role only when it binds with their receptor. So we wanted to know whether these cells are showing some expression of complement 5a receptor. So we measure this, and you can see as compared to control cell, disease cells are showing significant increased expression of C5AR1. So C5a interaction to C5AR1 on immune cells are known for causing their activation, which is mapped by upregulation of co-stimulatory molecule like CD40, CD80, CD86 on macrophages and dendritic cells, CD40 and CD40L on like lymphocyte, like T cells or B cells, which is known. So we wanted to know what could we measure in microglial cells that can suggest that these microglial cells are activated in disease state. So there are a couple of thoughts here, because it is well known that IBA1 is generally found on the microglial cells, but in like a normal level. But in disease state, it has been shown that IBA1 level significantly increased. So we wanted to check this marker. So we wanted to know whether there are some changes happening in compared to the disease. So you see, as compared to control cell, disease cells are showing significant increased expression of this marker, suggesting that this, in the absence of ADSL enzyme, microglial cells get activated. And that could be because of the excess SAICAR accumulation. So, and the second thing we wanted to know, if this is the case, whether these cells, these neuronal cells, are producing some pro-inflammatory cytokine. We generally measure TNF-alpha, IL-1 beta, IL-6, and IL-18. And what you have seen as compared to control, disease cells are exhibiting increased expression of TNF-alpha, IL-1 beta, IL-6, and IL-18, which is known for involving their role in several neuroinflammation diseases. So based on these data, I come up with this conclusion that because of the excess SAICAR accumulation, microglial cells or neuronal cells gets activated, their complement system gets activated, they release the C5a. C5a goes and bind with their receptor or neighboring neuronal cells and causes the cell damage. And that could be the reason that we see the neuronal cell death. We also generated some of the data where we used the same control and disease cells and treated them both in the presence and absence of complement 5AR1 inhibitor, which is called the PMX53. And interestingly, we've seen that as compared to disease cell, the treated cells shown significant improvement they basically protected the neuronal loss, and they also downregulated microglial cell and pro-inflammatory cytokine production. But we are still confirming those findings because that is really, really very exciting to me. And all data that you've seen here is the pooled iPSC from control human. So if we can get iPSC from the patient, because that was something that we had in our capacity, so we can work on it. But that would be really good idea to use iPSC derived directly from the patient, ADSL deficiency disorder patient. And we can also use the mouse model of this disease to see whether targeting complement at various steps can reduce, slow down the disease process or stop the disease process. So if, for example, if we will be fortunate and we see, yes, this is also the case with the patient cells and the mouse model, we can repurpose some of the drugs, for example, the avocapan, which is right now in the third phase of the human trial, and nicotinamide, which is already in clinic for treatment of these abnormality, like PNH, it has anti-AChR antibody and NMOSD. That's all I have to say today, and I'm happy to take any question.
That is great, Dr. Pandey. Thank you so much. So this is a really, especially for our families that are joining today, this is a really new kind of set of science. At least it was for me when I started reading it. So it's okay if you don't understand a lot. We're gonna dig in to this over time. What are any questions anyone has for Dr. Pandey?
I have
Otherwise, you guys can shoot your question to my email.
Yes.
I have one question, if that's okay. You hear me?
Yeah, we hear you.
Okay. Yeah. So thank you. Nice talk. That's really interesting. You said that ACAR interacts with this complement system, right?
Complement factor B.
Only with factor B. Is that what you said?
Right.
Okay.
Oh,
All
I
right.
mean,
I guess
only this is the data available. That could also possible that it may affect other component of the complement system, but this is something that I see available in the literature because it suppress the TNF alpha induced complement activation.
okay. Yeah, I need to review. I'm not like real familiar with complements. And so when you say the different parts of it, it doesn't automatically come to mind. I don't just know those. But I was thinking about, this idea that, lack of ACAR versus too much SAICAR could both play a role here.
Right.
Yeah. Thanks.
In terms of family perspective, we've talked a lot in our group about slow wound healing, children having a much harder time than their typical peers recovering from basic illness. As a matter of fact, I did run a Facebook poll. I didn't have a place to put it in my presentation, but I asked how many folks had had children hospitalized in the last 90 days. And it seemed, and let me not guess, but it seemed as though there were as many people in the hospital for rhinovirus as there were for seizures. And the rhinovirus is the common cold. So what we're seeing are children that are going into the hospital for very, very basic things that they are not able to overcome. And this has been something we've been talking about for years as a patient group. Why is this? Why do we see these amplified reactions to what other typical peers are able to clear quite quickly with maybe just a runny nose, and our kids are in the hospital, So this is, and as Wendy said, and I said too, the complement system and the immune system is totally never been, I don't think, ever really been talked about in terms of ADSL deficiency before. So it's certainly taking me a lot more research to understand, but it's very exciting. And I think it really we see the effects, potential effects. I don't want to talk about this like settled science, but we potentially see these effects of what you're talking about within the group. Can you talk a little bit about your lab?
So basically, we are looking the role of complement in various lysosomal storage disease, such as the Gaucher disease. So Gaucher disease happens because of the GBA1 defect. It is exactly the same like this is also the autosomal recessive disorder. So this gene is responsible for encoding an enzyme called acid beta glucosidase. The major function of this enzyme is to catalyze very complex set of lipid, which is called glucosylceramide. So glucosylceramide convert into the glucose and ceramide, which is vital. We need it for several of our metabolic activity. But because of this defect, when excess tissue accumulation of glucosylceramide occur, majority of the immune and neurological cells feel insulted. And in response to that, they produce pro-inflammatory cytokine and target different organs, not only the brain, but liver, spleen, lung is well known. And these patients also have very high incidence for developing B cell malignancy. And when these patients get older, they develop Parkinson's disease. So this is the thing. So in my lab, we nailed out how excess accumulation of this lipid actually causing that much inflammation. And there we find out the role of complement. So what happened in this disease, we published this in 2017 in Nature. So what happened in this disease, because of the excess accumulation and over the time, because of the natural cell death, they release into the circulatory system, B cells get chance to interact with this molecule many times. They lose their tolerance, they get activated, transdifferentiated into the plasma cell, which is known for producing antibodies to the molecule where they are interacting. So here the molecule that they are interacting is the glucosylceramide. So they produce antibodies to glucosylceramide. And these antibodies go bind with this glucosylceramide, make the complex. And this complex trigger the classical pathway of the complement activation system. So they release the C5a, and C5a interact with the receptor and cause all this damage. To prove this, we did the backcross with the C5a receptor knockout mice and the Gaucher mice, and developed both C5a are sufficient and deficient disease mice. And we also use the pharmaceutical approach on human specimens. So in both the cases, what we have seen when you knock down or you do the pharmaceutical blocking, there is no inflammation. So that kind of study we are doing. And then, as I said, because this disease, over the time, when the patient get older, they develop the Parkinson. So I also started to work on the evaluating the role of complement in Parkinson disease. And I had the funding from Michael J. Fox Foundation. Paper is in my table. Soon we will publish that paper. And then I also started to look the impact of this pathway in other lysosomal storage disease, such as GM2 gangliosidosis, particularly the Sand-Harp disease. Very interesting data we got. So I compile all those things and make the proposal for NIHR 21, which is scored pretty good. It is likely to be funded. So this is all tool that my lab is doing. The role of complement in inflammation in various diseases.
Amazing. So I'm going to ask a question I'm sure you won't have the answer to, but I want to think in terms of clinical care and what I imagine, what I'm wondering and what I imagine other families are wondering, which is there anything known that can support the complement pathway that is not a pharmaceutical? So let's say, have you looked into the role of diet, we're looking at neuroinflammation, and there's, there are anti-inflammatory diets. Do you suppose there's anything a family could do in the absence of a proof of the, full proof of concept and full workup of this to support their child's immune system in the meantime?
Um, my I don't have any idea whether there is some diet that can control complement activation. But of course, uh, when you take the
Pete, how are you? Nice to see you.
Yeah, good to see you.
Uh-oh.
What's going on? What are you doing down here?
Oh, just
when you
looking
take
at
the
some-
good
Nice.
ingredient,
One moment.
is
I'm gonna-
How things going?
Good.
the
Good. How's
vegetables and fruit, I think that would be useful. But I cannot really suggest any diet that can control complement activation.
I didn't think you would be able to, but I wanted to ask the question
Right.
because we have been working on, what would be the best diet for our children, and we've just been doing this through trial and error and seeing, what works over time. But dietary considerations are something that we are absolutely interested in. Yeah, and Carmel is saying, what about a diet that can reduce inflammation? We also notice a lot of our children in the ADSL group have lactose intolerance. And so, I'm not sure if this is all connected, except I think anything we do to inflame the body, if there's already, and you talk about neuroinflammation, which maybe one question I have is, do you feel that neuroinflammation is different from other types of inflammation in the body, or is it all sort of the same? Does that make sense?
Yeah, so neuroinflammation is different because it is happening in the brain, where a lot of sensitive cells are there and they are controlling huge function of your body. So neuroinflammation is really, really very critical when you compare to inflammation happen in your visceral organ like liver, spleen, or lung. They have one kind of function, but brain is controlling all body functions. So
Mm-hmm. Yeah,
that way it could be different.
yes. Wonderful. Any other questions for Dr. Pandey? This has been really, really wonderful, and I'm so excited to see where this leads you, Dr. Pandey, and where it leads our group. Thank you for being here. And
Thank you for listening me.
Oh, of course. We're so happy to have had you here.